Good morning students. Today you are going to study about fertilization. First you can recall what I have taught already. Pollination. Pollination is the transfer of pollen grain to the stigma of the flower. So pollination is the transfer of pollen grain from the anther to the stigma of the flower. There are two types of pollination. What are the two types? Yes, self-pollination and cross-pollination. Among the self-pollination and cross-pollination, which one is better? Yes, you're right. It's a cross-pollination because it produces new varieties and healthier offsprings, etc. Let's see fertilization. Fertilization means the fusion of male and female gamete. The fusion of male and female gamete is called fertilization, otherwise called syngamy. What is that? Syngamy. The fertilization in angiosperm is a double fertilization type. Double fertilization type. Then what are the events of fertilization? Some stages are there. Let's see one by one. Germination of pollen to form a pollen tube. First one, germination of pollen to form a pollen tube in the stigma. Then the growth of the pollen tube in the style. The growth of the pollen tube in the style. Next, direction of pollen tube towards the micropyle of the ovule. Next, the entry of pollen tube into one of the synergids of the embryo sac and discharging male gametes. Next, syngamy and triple fusion. So all the events from pollen deposition to entry of the pollen tube into ovule, all are called pollen pistil interaction. What is it? Pollen pistil interaction. See, the transfer of pollen grain from the anther to the stigma of the flower, that is called pollination. So after the pollination over, the pollen deposited on the stigma of the flower. The pollen deposited on the stigma of the flower. Then as the suitable pollen fall on the suitable stigma, the first visible change in the pollen is hydration. What is that? Hydration. Then during germination of pollen, its entire content move into the pollen tube. Entire content will move into the pollen tube. Then the extreme tip of the pollen tube appear hemispherical and transparent when viewed through a microscope. This is called cap block. What is that? Cap block. So the pollen grain fall on the stigma, then it gets hydrated and it start germinating and it will form a pollen tube. Then all the materials, that male gamete tube, all that it will enter in, discharge into the pollen tube. Then at last the tip, it will be called as a cap block. After the cap block formation, there will be no growth of the pollen tube. The pollen tube stop its growth. Then the pollen tube in style, there are three types of style: hollow style, solid style, then semi-solid style. First one, hollow style. Hollow style, otherwise called open style. Here it is found in the monocots. It's a hollow canal from stigma, hollow canal from the stigma to the base of the style, and it is lined by a single layer of glandular cell. Both the sides, it is lined by a single layer of glandular cells. Next one, solid style. It is otherwise called closed type. It is found in dicot. Here, instead of the single layer of glandular cell, it consists of a central core of some special cells. That special cells are called transmitting tissue. Transmitting tissue. Next one, semi-solid style. Next one, semi-solid style. It is half closed. It is the intermediate type of hollow and solid. Then the next one, how the pollen tube entering into the ovule. How the pollen tube entering into the ovule. There in three ways it is entering into the ovule: porogamy, chalassogamy, mesogamy. If it is entering through the micropyle, this part, it is called porogamy. If the pollen tube entering through the chalassal part, it is called chalassogamy. If it is entering through the integument part, it is called mesogamy. So based on the part it is entering, it is three types: poro, chalasso and mesogamy. Let's see the entry of pollen tube into the embryo sac. Entry of pollen tube into embryo sac. Here the growth of the pollen tube towards the ovary, ovule and embryo sac is all due to some chemotrophic substances. Some chemical substances, it is attracting the growth of the pollen tube towards the ovary, ovule as well as the embryo sac. Here one structure that is very important for the entry of the pollen tube into the synergid, that is obturator. It's a structure which guides the pollen tube towards the micropyle of the ovule, That structure name is called obturator. After reaching the embryo sac, a pore will be formed in the pollen wall through which the content, two male gametes, vegetative nucleus and cytoplasm are discharged into the synergids, and the tube nuclei will be disorganized. So after reaching the embryo sac, a pore will be formed. Then through that pore, the two male gametes as well as the cytoplasm, then tube nucleus will be enter into the synergids, then the tube nucleus will be disorganized. Then what is double fertilization and triple fusion? See, scientist S.G. Navashin and Guignard observed in Lilium and Fritillaria, both male gametes are involved in fertilization. Two male gametes also involved in fertilization. So male fusion of male gamete and the female gamete egg, that is called syngamy, otherwise called fertilization. Here the second male gamete fused with the polar nucleus results in the formation of primary endosperm nucleus. This is called double fertilization. It is very unique for the angiosperm. So two male gametes also involved in fertilization. So it is called double fertilization. And here the second male gamete and the polar nucleus, that polar nucleus is diploid, and resultant, the primary endosperm nucleus is a triploid. So this gives a triploid nucleus. So we can call this as a triple fusion. What is this? Triple fusion. So one male gamete fused with the egg result in the zygote. That zygote will develop into embryo. Then another male gamete fuses with the polar nucleus, results in the primary endosperm nucleus that develop into endosperm. Next we can see the post fertilization changes. The events after fertilization, I mean the endosperm, embryo development, formation of seed, fruits, are all called post fertilization changes. You know what is meant by fertilization: double fertilization and triple fusion. Now we can see post fertilization. So the parts before fertilization and after fertilization, it is given that sepals, petals, stamen, style, stigma. Usually it will wither and fall off. Then the ovary will become a fruit. Then the ovule become a seed. Then the egg will become a zygote. Then the funicle, it will be the stalk of the seed. Then the micropyle of the ovule will become a micropyle of a seed. Then the nucellus, the central part of the ovule, will become a perisperm. Then the outer integument of the ovule will become a testa of the seed, outer seed coat. Then inner integument will be the tegmen. It is the inner covering seed coat. Then the synergids, antipodal cells all will degenerate. Then the secondary nucleus will become a endosperm. Then the zygote will develop into embryo. So which part will become a fruit? Yes, it is a ovary. And which part will become a seed? It is a ovule. Then which one will become an endosperm? It's a secondary nucleus. So the events after fertilization, the endosperm, embryo development, formation of seed and fruit, all will be called as a post fertilization changes. Let's see the endosperm. Endosperm, the primary endosperm nucleus, it's a resultant of the second male gamete and the polar nucleus. The primary endosperm nucleus undergo mitotic division and it will result in the endosperm. But it is developing before the zygote start to divide. Before the zygote start to divide, the endosperm started to grow. It's a nutritive tissue and regulatory structure that nourishes the developing embryo. So endosperm is triploid condition. It is a nutritive tissue that nourishes the developing embryo. It's a result of the triple fusion. Here there are three types of endosperm: nuclear endosperm, cellular endosperm, helobial endosperm. Nuclear, cellular, helobial. First, nuclear endosperm. Here the primary endosperm nucleus undergoes several mitotic division without cell wall. Without cell wall. So here a free nuclear condition. A free nuclear condition will be formed in the nuclear type of endosperm. Example, coccinia. Example, coccinia. Here no cell wall formation, free nucleus condition. Second one, cellular. Here the primary endosperm nucleus divides into two nucleus, then it is immediately followed by a cell wall formation. Immediately followed by a wall formation. Then many divisions will be there, all also followed by the cell wall. Here example, helianthus. Example, helianthus. Next one, helobial. Here, the primary endosperm nucleus move towards the base of the embryo sac and divide into two nuclei. The cell wall formation takes place, leading to the formation of a large micropylar part and a small chalacal part. So the micropylar part, it will divide and several free nuclear division will be the several free nucleus will be formed, but chalacal part it may divide or may not divide. Example, hydrilla. Example, hydrilla. Then in some plants, endosperm with irregularity and unevenness. That type is called ruminate endosperm. So if the endosperm with irregularity and unevenness in its surface mean, that is called ruminate endosperm. Example, areca. Here the functions of endosperm. It's a nutritive tissue for the developing embryo. Then majority of the angiosperm, the zygotes divides only after the development of the endosperm. I said first itself. Then it will regulate the precise mode of the embryo development. It will regulate the precise mode of embryo development. So these are the functions of endosperm. Next we can see what are endospermous seeds and non-endospermous seeds. Here, endosperm may either be completely consumed by the developing embryo. In such cases, we do not find endosperm. So, the seeds without endosperms are called non-endospermous seeds, otherwise called X-albuminous seeds. So, the seeds without endosperm, that is non-endospermous, otherwise called X-albuminous. So, the seeds with endosperm are called endospermous, otherwise called albuminous seeds. So these seeds with endosperm supply nutrition to the developing embryo during seed germination. Here example, paddy, coconut. Then non-endospermous example, pea and groundnut. Here I will give 5 mark question. Study and write as self-test. Thank you.